So, Jackie, thank you so much for coming on with us tonight. Would you mind giving us a brief introduction of who you are?
Hey, my name is Jackie Litzgus. I've been a professor at Laurentian University, a professor of biology at Laurentian for just over 20 years. My expertise is in the biology and conservation of reptiles and amphibians, and most of my focus has been on turtles, but I also do quite a bit of work with snakes. Every now and then I dabble in frogs and toads.
How did you get into working on snakes, toads, lizards, other reptiles? It just sounds like that's such a unique field, and it's not something that we regularly hear about when it's usually like yeast, fungus, or a fly. How did you get into that field?
I was actually super fortunate. What I did as a kid became my career. So when I was a kid, I have two older brothers. One of my older brothers, who's now actually a high school biology teacher, he dragged me around swamps and creeks, and we caught things. We weren't allowed to have regular pets like dogs and cats, so we caught snakes and we brought those home, or we caught toads and we brought those home, and we'd keep them for like a week and feed them, and then let them go, and then go find another one. So I have this super rare privilege to have turned my childhood fascination into my career, and it's so cool now when I talk to students who are in my courses. Like I teach a course in herpetology, which is the study of reptiles and amphibians. And students are like, wow, you can actually do this for a career? Like the stuff I love to do, running around in a swamp or a creek. I'm like, yes, you can. And I'm living proof. So it's fascinating. And it's out of a passion for the animals that I get to do research on them and learn even more about them. And they have so many stories to tell. So it's a great opportunity, and it's constant learning and excitement for me anyways.
So it's really cool. Like, how did you realize, or when did you realize that it's one of your favorite animals, like reptiles, to work with? And then at what age did you feel like, this is what I want to do?
So I was pretty naive when I went to school. So I mean, I went to university. I went to high school, went to university, and that brother that I was talking about, he was interested in animals and such too, right? So he went to the University of Guelph for his undergrad, and so I thought, well, I'm going to go check that out too. That's what he did. So I ended up going to that same school, which had a specialization in wildlife biology. But even while I was there, I wasn't entirely sure what professors did, and that they could, in addition to teaching, that they did research and all these other things. Like I didn't really understand that until say like maybe third or fourth year of my undergraduate degree, that I finally realized that there's a job that you can do where you get to do that kind of stuff. And the people that were teaching me the laboratory demonstrations at university were actually these things called graduate students that could go on and study research. Like I didn't understand that. Neither of my parents actually finished high school, right? So like I came from a family that didn't have that academia background at all. It was very so I said two older brothers. All three of us went to university and then went on to advanced degrees. And my parents are like, what the heck? Like, how did we produce these children? But we all did. We all are like really independent, motivated kind of people to go on and do that kind of. So anyway, once I learned that it was possible to do that as a career and to do a master's degree, then that was like a whole transition for me. Everything changed at that point when I realized that you could pursue that as a career. It wasn't like I was 7 or 8 years old going, I want to be a professor when I grow up. Like I didn't need like that. I wanted to be a wildlife biologist of some kind, maybe a vet. Like I toyed with that idea for a little bit, but it would be like an exotics vet, not a, dogs and cats type vet. But you have to have really, really good marks. And I didn't have really, really good marks. So that wasn't really an avenue for me.
What was the first animal that you fell in love with in terms of like that driving you towards that wildlife conservation? Was it hunting for frogs with your brother in the backyard?
It was probably hunting for a species of snake called milk snake. There was a field where my brother and I could ride our bikes to it, and we would go on weekends and flip over, old pieces of wood and whatever, and we'd find these beautiful milk snakes. They're like 4 or 5 foot long, gorgeous snakes, and we were just amazed that this thing that looks so exotic is right there in the field, by our house. And then we discovered all these other snake species that live there too. So it started with snakes. And then I would say the second creature was toads. We would find toads all over the place, and we'd go under the street light on our street, right? And all the bugs would be out, the toads would be out. So that became our thing. And then we got into turtles a little bit, but turtles are really hard to keep. Turtles are terrible pets. Nobody should ever keep a turtle as a pet. They're dirty and they require all kinds of water and lighting. And anyway, so we would go find turtles, but we didn't keep turtles. But snakes was definitely the first thing. And I loved how they looked, how they felt, their variation in colors and patterns. And yeah, so that was definitely definitely the fascination snakes started.
Really cool that you're able to do that with your brother too. It sometimes is like, you hear brother sister relationship and it's like, no, that's gross, like don't touch the animal or something, but it's cool that you're all able to share in that.
Yeah.
When you ended up, so you went towards to get a master's degree first, how does one go about navigating that and parlaying it towards reptilian research, or were you not at reptile research yet?
I was. So there's another interesting serendipitous story, and it involves my brother again. So when we were both at Guelph, they had like a career board, right? And it posted different jobs. And he found a job posting there to teach biology. He knew he wanted to teach biology eventually, to teach biology at a kids' canoe tripping camp on an island in the middle of Georgian Bay. And so he went and he took that job for a summer. And he came home after the summer, and I'm like, wow, what was that like? Did you like it? And everything. He was like, yeah, it was really great. And then typically when you're a biology teacher at that camp, you do it for 2 years. You're the junior biology teacher, and there's a senior teacher, and then you go back the second year and you're the senior teacher, I think, and hire a junior. So the lady that was the director of the camp called our house and asked for my brother. She wanted to see if he was going to come back to the camp. He wasn't home, and I said, well, I'm his little sister. Can I come and teach biology at your camp? So it was totally this, I'm holding a phone. Like, does anybody know what a phone means? Would I do that anymore? I should hold this instead. Anyway, so it was just, I was gutsy, and I'm like, it sounds really cool. I want to try it. And so I went for an interview. She hired me. My brother and I went back, and we were this brother-sister biology teaching team at this canoe tripping camp. And at that camp, there was a building called the Bug House. And that was the building we taught biology program out of. And in that building, I found this old binder with all of these data sheets in it. And they were like all mildewy and gross and stuff. I opened them up, and they're records of turtles that were captured near the camp by previous biology teachers. And then we went out and we found those exact turtles. They were individually marked and then recorded in these data sheets. So I'm catching these turtles. It was like, for me, opening a treasure chest, right? I'm like, oh my God, and we're catching them. And so that is what made me realize you could do these kinds of long-term data collection. You could understand the animals. And then in first year in my undergrad, I had a professor who was a turtle biologist who taught the introductory zoology course. So I went to him and said, I found these records and I found this turtle. He's like, wow, so that's how I got into a master's, is by this serendipitous, moment of going to teach biology at this camp, finding this treasure trove of data, and then finding the turtles, and then approaching this prof that I knew worked on turtles and said, hey, can I do that thing called a master's with you? He's like, okay.
That is so cool. Like, and your brother too, like what a cool brother to be able to have a brother-sister team to go hunting for turtles.
Yep.
Did that data ever become a paper?
Those data became my master's research. So with my brother, we contacted 2 people whose names were on the data sheets to see if we could get all of the data, because it spanned like years and years. And we contacted them, and they said yes, and they handed us all of the data. And then I pursued that as my master's degree, and I think I published 4 or 5 papers out of that data set. And then I added to it. I added a radiotelemetry study to it. So there's marking individuals and monitoring them over time. But then I outfitted some turtles with radios to understand their spatial ecology too. So it resulted in quite a bit of work. And then 10 years later, one of my first grad students did her master's on that same population 10 years later, because there was a shift in how the turtles were using the environment. We were trying to discover what caused that shift. So then 10 years later, I had my own student go do a master's on that, and she published a couple papers. And now actually, I still go back and survey that turtle population every year in the spring, the beginning of May. And I've done that for, I don't know, since 1991. That ages me. But anyway, that's how long I've been surveying that population of turtles.
Is the camp still there?
It is.
Are you like camp royalty there?
So when I go do my survey, the camp's not open yet because it's a kids' summer camp. It's only open basically for July and August. So I go in early May and the camp's not open yet or anything, right? So
that is such a cool story about getting into the science and the master's degree as well. So what was the research question on those populations? And how does one formulate a research question using turtles? Because it's, again, very, very different from the research that a lot of us are familiar with, where it's at the bench, you're extracting, you have them in little tubes. Like, you don't have these turtles in tubes. So how does one actually go about formulating a research question when studying this kind of animal?
Yeah, and that's a really good question, because there's definitely a difference between, say, a Drosophila melanogaster as your model system versus turtles. So if you're addressing a genetics question or an evolutionary question, you pick a model system that suits that question best, like fruit flies, for example. In my case, it's a bit different. My research interest is actually the animals themselves, right? So my research questions are focused on understanding some particular aspect of those animals so that I can understand their ecology and then contribute to their conservation. Empirical data to support conservation. That's my research agenda, I would say, right? So it's all field-based research. We're out in the field monitoring the animals in their natural environments. And so the research questions for that first project, it was the species I was working on was not well known. And part of it was just understanding their biology and their sort of natural cycle. Where do they hibernate? Where do they mate? What do they eat? Didn't get at that very well. Where do they nest? Like, where do they lay their eggs? And those things are all important. Those are spatial ecology aspects that are important to identifying where the animals go, when they go there, and what they're doing. Because then if we can understand that, we can conserve that system so that they can continue to do that. So one of the things about turtles is that they live for a really long time. So snapping turtles may be like a common species that most people have seen, right? Probably 100 years, pretty easily for an individual to live, but maybe even longer. Some models suggest they live for like 200 years. So understanding how they use landscapes is super important to their conservation if we want to see them continue to live for those kinds of durations, right? So one was to understand that ecology, that general, what habitats do they need, when do they need them? But then there's a more, so that's sort of the applied science, right? The conservation related, but I'm also interested in fundamental science with respect to things about the life history of animals. Like, what are their age and sizes at maturity? How big are their clutch sizes? And how does that impact population viability into the future? So it still has a bit of a conservation twist, but it's grounded in this fundamental aspect of their natural history, those attributes that lead to their survivorship, but attributes related to their fitness, right? Survival and reproduction and stuff. So I was also interested in that. I wanted to understand the life history of these animals, because those features actually change by population. So I was working at the northern range limit for the species, and then things change though, because environmental selective pressures change, right? So for example, age at maturity is delayed in the north because turtles they're cold-blooded, which is not the accurate term, right? But they're ectotherms, so that their body temperature mimics the environment. So where it's cold for a long time, it takes them longer to get to maturity versus down in the south where it's warmer. And so that affects all kinds of things about their life history. So I was interested in those kinds of patterns and then how we use that to inform conservation of these animals.
Really an imposing question. It's kind of scary almost, right? Like, if you know nothing about this animal, like you were saying, like what they eat, how fecund they are, what their mating preferences are, like that's a really, really big undertaking. Like you said, it's very critical for maintaining that species, but just seems so foundational too that it could be quite scary to get into.
It was a big project. I had these, I had those long-term data, and then so my master's took me 3 years because I did have a lot of questions. And to be frank, I lacked focus because I didn't go in with this one very narrow research question or hypothesis. I went in going, oh my God, I want to know about these animals. And my supervisor was very hands-off. So I went in to learn about the animals and ended up, it took me 3 years to get my master's instead of the average, 1.5 to 2 years. But that was okay with me. We ended up with really good financial support to do the work from the World Wildlife Fund. They gave us a third year of research funding to learn more. Like, they felt it was important too. So I wasn't in a rush. I mean, there was lots of life ahead to plan. I was happy to just be out in Georgian Bay chasing turtles for another season.
For a question from Chad, do they migrate like to warmer climates to grow fast, the ones you were studying, or are they just always endemic to that one area?
No. So they occupy what's called a home range. They have a very defined space, 3 hectares, 4 hectares approximately that they live in. But within that, they do migrate. So they have seasonal migrations within their home range. So they have a certain spot where they will hibernate. They'll exit that hibernation spot and move to a place to mate and feed, move to another place to lay their eggs, move to another place for their sort of summer hangout, and then they'll go back to the hibernation site. So they have very predictable cycles. In fact, there's one female from my studies, that long-term study, my master's, and I brought one of the original biologists with me to the field site about 2 years into my master's. And we walked over this big rock outcrop, and there's this beaver pond, and that's where the turtles live. And female number 2 was basking on a log there. And I said, Mike, biologist, I said, do you recognize her? He's like, what? Like, that's female number 2. You marked her in 1977. He's like, man, she was on that log back then too, kind of thing. Like, mean, it wasn't that precise, but pretty close, right? Like he was like, oh my God, she's right there. Like, yeah. And I said, she's here all the time. I see her every year, kind of thing. And so they're tenacious and they show fidelity to these systems. And so that's another aspect of their conservation that's so important. I was talking about knowing where their seasonal activity stuff happens, because we need to preserve that whole thing so they can complete their annual cycle. So one of the huge problems for these animals is roads, right? We build roads that cut right through the middle of their cycle, and then they live for decades and decades, and the road shows up in the middle of their lifetime, and now suddenly they have to contend with this gauntlet that they have to run every year, right? And females are attracted actually to the sides of roads, the road shoulders, to lay their eggs, because it's really good diggable soil, right? Like the gravel sand mix on the road shoulder. Sometimes females are attracted to that nice open sun exposure too, a good place to put their eggs. So they lay their eggs there. So that puts the female at risk of getting hit by a car. And then it puts the nest at risk of pollution and runoff from the road, maybe compaction because if a car pulls off onto the shoulder. And then when the babies hatch, if they do, then they run the gauntlet of cars flying back. So roads have become one of the most significant threats to turtles because of that great longevity, because of that fidelity to their migration patterns. And then suddenly, bam, we stick some piece of infrastructure in the way, and it's detrimental. And because of this very slow lifestyle that turtles have, as soon as you take out an adult, it causes a lot of damage to the population. The persistence of the population tanks when you take out adults, especially adult females.
So it's a really cool insight about just like the importance of the conservation element to them as well. Like you were saying, it's not a particular single zone to look at, but rather the different zones that they migrate through, and then taking into account what area to preserve. You all know why they do this migratory behavior within the zones throughout the season. Like, what is it about this zone where I want to breed, and this zone where I'm going to hibernate, and this zone for when I'm waking up?
And right there is one of the most important research questions that people who study freshwater turtles are looking at, right? So why do they go there? So we know, for example, where they hibernate. So up here in Ontario, right, the turtles spend about half their lives hibernating, right? There's about 5 to 6 months every winter where they have to go hibernate. And my research team has actually done studies on this on multiple species to try and figure out what it is that the turtles are choosing about a particular place when they go hibernate. And we know a few things, and it depends on the species a little bit, but we know that they want to be as close to freezing as possible without freezing. So they want to be just hovering above 0 degrees Celsius. And we think that's because they're cold-blooded, right? Like I said, they're ectotherms. That lowers their body temperature to just above freezing, which means their metabolism is exceptionally low, which means they can survive under a lid of ice. They have lungs under a lid of ice without going up for a breath of air for up to 5 or 6 months. Like, can you imagine going that long without a breath of air when you breathe with lungs? Like, no endotherm mammal could ever pull that kind of stuff off, right? But these turtles, they slow down so much. Their oxygen demand is really low. Their metabolism is really low. They don't need to use energy. They're not burning anything, right? So they can just hang out really cold, chilling out, literally at the bottom of a pond for a long period of time. So we know that being as cold as possible is good because then it really slows that metabolism down. We also know that they're choosing places to try and avoid predators. So when turtles are hibernating and cold like that, they're very slow. They're lethargic, right? So they're easy prey for like an otter or something like that. So they try to choose spaces under the ice where this, and again, these are hypotheses that we have based on what they choose, but that minimizes that headspace of open water so that it's harder for predators to get in. We also know about like dissolved oxygen in the water. So some species really try to pick spots that have more oxygen, and they can actually uptake oxygen from the water column across skin surfaces to some degree. Like they can use buccal pumping. They can pump oxygen across the vascularized surfaces in their mouths a little bit. But for the most part, they just don't need that much because they're so cold. So that's hibernation. We've actually worked on that hibernation aspect of things, which is challenging because you're following animals at minus 20 degrees Celsius, trying to figure out where they are with gear, with rib telemetry gear.
Yeah, that's what I was going to ask. Was like, I know we're so rabbit holing already with like, the journey.
Sorry, I go off on tangents quickly.
No, I think it's just the excitement of the research too, right? Like, so could you talk a little bit to us? Like, how does one actually track the hibernating turtles. And then do you, like, we talked about metabolism too, is there any genetics in the field where there's like transcriptomes done at this stage yet to figure out like what are the genes being turned on and off that can allow this animal to, live 6 months underwater? Like, a community member in chat just said, I struggle after 30 seconds. And just imagine like if we could figure out the secret for that kind of like cryostasis.
Yeah, and that I don't know. I don't know if people are looking at that aspect of how they overwinter, the like deep molecular physiology of it. I don't know. People have looked at the physiological ecology of it, and like sometimes when turtles are hibernating like that, they'll switch to anaerobic metabolism. So then lactic acid is the byproduct, and that leads to acidosis in the blood, and that can be lethal after an extended period of time. So research has been done on that kind of aspect, but I don't know anything about transcriptomics and if that's been researched. It may have been, and it's just outside of my expertise. But monitoring the animals so we use a tool called radiotelemetry, and that is radio transmitters. A little unit is glued to the shell of the turtle. Every turtle has a different radio station, a different frequency. And then we use an antenna, a 3-prong Yagi antenna it's called, and it's attached to a receiver. And in the receiver, it's just like the radio itself. You dial in what frequency of the turtle you're trying to track, and then you use the antenna, which is directional. And so wherever it's loudest, that's the direction of the animal. And so then you can radio track to the animal under the ice. So like we stand on top of the ice and we radiolocate an animal down below us, and we know that that's where it's hibernating. And then we can collect, like sometimes we make a hole in the ice and we collect information on temperature, dissolved oxygen. We try to get a visual on the turtle if we can. That's not always easy to do. But yeah, so radiotelemetry has been a real essential tool for understanding the sort of secret lives of turtles, because you see them basking, early in the spring, and then they kind of disappear and you don't see them very much. So without these instruments, we wouldn't know very much. I mean, there's satellite tracking too, which is pretty expensive, and it doesn't work super well for turtles because it doesn't work really well underwater. But VHF radio transmitters is what we use, and they work really well, really well.
That's really cool that you have like then the ranges that you can get, and then monitoring them too. So when you go and dig a hole in the ice, let's say, and then you're zooming in, so presumably you don't actually have to physically interact with them, so it doesn't affect their hibernation status, but you can get all this data around them. And in theory, right, like your master's student who went back to the same location, like you could have someone go back to that same location in 20 years too and keep tracking. You said what, up to 200 years, right, for a snapping turtle of going, like that's an amazing treasure trove of data, like you were saying. It's super, super cool. So I can see how maybe one would be smitten with the field, right? So how does one then parlay from going from the master's degree into the continuing education, focusing on reptile research? Because it seems like, at least from our experiences, that, it's kind of niche and there's not a lot of people studying this particular research question. So how were you able to continue studying it?
Yeah. So that really depends on finding a professor, somebody who can be your supervisor who has that expertise. And so in Canada, there's some small complement of professors that are herpetologists or who at least use reptiles and amphibians as their model systems. And maybe they're even asking more genomics type questions, but they still have this sort of love of the herps. And so I belong to a scientific society, the Canadian Society for Herpetologists, and we have a website, and one of our things that we do on the website is to list Canadian herpetologists, Canadian professors with whom you can conduct research. And so, I mean, it's a small group of us. Like whenever I go to these Canadian Herpetological Society meetings, it's like a family reunion because most of the people there have been in my lab at some point. So it's kind of a fun. It is like a family reunion, and you get to know who the herpetologists in your field are just by reading papers too, right? So every scientist, the first thing you do is read a whole bunch of papers, right? When you're setting up your research question. So then you start to become familiar with these names. And like I said, there's not a ton of people. So then you know who to reach out to if you want to continue your studies.
What was your research question for your graduate work? Like after the master's degree, and how did you decide on that? Were you able to continue with the same site study as in your master's?
No.
Or was it brand new organism? Like where did you?
No. Okay. So it wasn't a new organism. It was actually the same species again. So sorry, I'm losing light here. I'm just going to lighten up a little. So I had an interesting path for sure in getting to where I got to. So I'm backing up even maybe a little bit. So for my undergraduate degree, I started first year. I I had a scholarship. It was really, everything was great. But then my marks tanked. So I had really high marks in high school, got a scholarship. And then first year university, I think I was too young. I think I wasn't mature enough to do it. And my marks tanked. So I actually dropped out for a year. And I worked in an office and I'm like, oh, this sucks. And so I went back to school and my marks went up. I never got my scholarship back or anything, but my marks went up and I had a new view on things, right? And I started to become interested in my education a lot more. And then I learned about masters and I did my masters, and that was really great. Then after my master's, I was like, I'm not sure what I want to do next. I know I love this research thing. I want to try some new stuff. So I actually worked as a research lab researcher in the States. I moved to Ohio and I became a manager of a research lab that studied freezing and overwintering of reptiles and amphibians. For 3 years, I did that position. I worked for 2 professors. I learned a whole bunch of new skills. We published many papers out of that work. So I got this really cool taste for another way of doing research. It was all bench work, almost all bench work. There was some some field work, but it was always to serve the purpose of the laboratory work. So I learned a lot of new skills, and it was interesting. And one of the other things I learned, and I think this is important, and I tell this to my students all the time, learn how you are as a person and your personality and how you interact with other people and what kind of person you would like to collaborate with. And it's good to know what you don't want. And so I really encourage my students to go to different places, to have the experience of different people and different places, because I think that's as important to your scientific growth as the science itself. And I have seen students leave science because of personality conflict, not because they were disenamored, they weren't loving science anymore. And I find that very sad. So I went and did this thing for 3 years doing somebody else's science. And I'm like, I don't like being controlled. I want to be in control of my own destiny. I need to go do my PhD. And so from my master's, I still had all these other questions. So I was telling you about the latitudinal patterns in things and life histories. And I was studying a population at the very northern extent. So I'm like, what do they do in the South? How does all of this change? How do all these life history attributes change in the South? How do their population dynamics change in the South? And what does that mean for conservation of this animal in the South? So I looked further south to find the most southern population of that species that I could, and then started reaching out to people to say, this is what I want to do. Will you let me?
So naive question, Jackie. How does one start to look at how south turtle is?
So because of having done what I did for several years, I knew names in the literature again, right? And there was one particular scientist who I adored. He's one of the grandfathers of turtle ecology in the US. And I knew he had dabbled around with turtles in the South. And so I wrote to him and I said, where do you think would be the most southern place I could go and still get a good enough sample size, right? If you get too far to the periphery range, you might only find a few individuals and then you've got no sample size to get any good data, right? So I picked his brain and said, how far south can I go where I'll have a large enough population to actually have good data? And so he suggested South Carolina. So then I started investigating South Carolina, and I reached out to people that might know about the turtles there, and then landed on a couple of potential locations. And then I wrote a grant proposal, and I can't remember who I wrote it to, but a small grant proposal to get travel money so that I could go and scout. So I did that, and I discovered the population, or I identified the population I wanted to work on. And then I wrote to universities and said, would you let me come and do this? So it's a bit backwards. Often that's not how a PhD works.
Yeah, it's like you did all the legwork, right? Like getting the grant for the travel, finding the population to study, and then identifying a university like, hey, okay, I've got this research question, I've got this population, I'm ready to go. Is that normal, like an experience for this type of work and with reptiles, or it was just you are an absolute badass and you were just like, I'm just going to do that?
No, it's not that. It's not normal, but partly why I did it was because for those 3 years that I worked in the lab as a lab manager, I realized a lot about myself. And that is that I like to be in charge of my own research. Like I don't enjoy doing somebody else's work all the time. And so if I was going to do a PhD, I wanted to be in charge of what I was doing. So when I pursued this idea, it was what I wanted to do. Even actually one of my supervisors at the job in Ohio said to me, you're just continuing on with what you did before. Why don't you try something new? I'm like, I've been doing something new for 3 years. I'm ready to go back to my roots kind of thing. Like, this is what I'm excited about, what I'm driven about. And so when I interviewed for PhD, I think I think I was being interviewed as much as I was interviewing them because I knew I needed to work with somebody who would let me grow my own project. Like I really wanted that idea. And if I was going to fail and crash and burn, then so be it. But that's what I wanted to try. So that's what I did. And then I interviewed with 2 different professors for PhD, and one of them would have paid me more money. He was going to tell me what my project was. And I'm like, nope, that's not it. And the other guy, I went out with the professor I ended up working with, Tim Mousseau. He's also a Canadian, expat Canadian who's down in South Carolina. And I went out with a bunch of the grad students when I went down for my interview to the pub kind of thing. And I asked, what's it like to work with Dr. Mousseau? And they're like, he'll give you enough rope to hang yourself. I said, that's my man. Like, that's what I want.
That's so cool. We've got a few questions from the chat about the journey. So I guess same parts. One is when you were looking for these other turtles, or even now if you're looking for certain turtle populations, you ask for citizen science like reports. Is that something that's beneficial to you in your line of work? And then the side question was, if you didn't get that grant money when you were doing the travels, what was your plan B?
Yeah. Okay. So I don't very much tap into things like INAT and stuff like that to look at locations. However, like I said, because the herpetology community in Canada is pretty small and the turtles are pretty restricted at where they are, kind of know the distributions of the animals already just from having been boots on the ground here for 3 decades. So in terms of citizen science, I mean, I work with communities. I do a lot of work with communities and grassroots organizations. I work with First Nation communities on their lands. I work with like biosphere reserve type agencies, and they have their own data sets as well. And so we pair with each other. So yeah, I think that's mostly how that has worked. If I hadn't got that travel grant, I probably would have just paid out of pocket to go investigate. So I guess I am naive, as I said, and that makes me maybe take some chances that other people wouldn't take. But I kind of went on a wing and a prayer. Like I wanted to do this, and I'm stubborn. So I was like, I'm going to figure out a way. And when you're stubborn, luck kind of goes your way sometimes, I guess. I don't know. Anyway, that like in that first grant, that was just a very small travel grant that allowed me to pay for my gas, basically to check out these possible sites. And then once I chose the place to go for my PhD and I ended up in South Carolina, that professor did not have any money explicitly for my project, but I wrote a National Geographic grant and I got that funded, and that paid for a lot of my research. And then I was able to partner with some other, I worked with Audubon and Nature Conservancy while I was there. So there was a lot of in-kind support. I basically beat the bushes, to find people that would help. I was even building my own radio transmitters for a little while, which was a pretty abysmal failure, but I tried. I mean, I don't know how to solder, but I learned, I tried to, I was trying to be resourceful and figure it out because it's what I wanted. Like when you really want something, you make it work, you find ways and you take chances on stuff. And I would like the whole serendipity thing. Like, what do you have to lose? I talk to students sometimes too about I met a high school student recently and she was asking me about herpetology because she's totally in love with that idea. And she's in like grade 12 and she's got a 5-year and a 10-year life plan. And I'm like, it's really cool to be organized, but have some flexibility. Like what if an opportunity lands at your feet, but it doesn't align with your 5 and 10-year goal? You might miss out on a chance that's really rock solid. So I mean, it's good to have a plan. I'm not saying that I hope her parents aren't listening to me right now because they're going to be like, oh, Dr. Litzgus, why are you telling her that? But I think it's good to take opportunities when they present themselves. And the other thing I tell my students often too is don't let money be the only thing that drives the show. Yes, you have to have money. I mean, yes, you have to eat. It's nice to have a car and a cell phone, but don't let that be the only thing that decides what your future is going to look like, because money doesn't make you happy. Doing what you love makes you happy, and the money just sort of falls into place, right? Like that's I've been super fortunate. So it's easy for me to say that everything worked out for me. Well, there were times when I couldn't make my car payments, but oh well, I figured it out. I tracked turtles.
I think you're the embodiment of chasing your dreams, right? Like that's at the end of the day, if it brings you joy, like you were saying, go for it. So you've gotten this now position in South Carolina, got the research question of the southernmost of this turtle. What were the findings? How are these turtles different from those northern turtles?
Behaviorally, it was pretty cool, actually. A couple of things. So up in Ontario, as I said, they hibernate for like 5 or 6 months of the year. In South Carolina, they didn't hibernate. They were active almost all year round. around. They got a little slower. There was an ice storm one winter when I was radio tracking, and I was a bit worried about the turtles because they weren't in places that like an Ontario turtle would pick to hibernate. I'm like, oh my God, they're going to freeze. This is going to be terrible. They were under leaf litter and in a bit of damp area, but they all survived. Nobody froze to death, so that was great. And the other thing, because the seasons are like in Ontario, very distinct: spring, summer, fall, winter, very concise, distinct seasons. And in South Carolina, it's really, really freaking hot, and then it's kind of cool, So that's kind of it. And so it means a really long growing season. And so whereas in Ontario, a female would produce a batch of eggs, a clutch of eggs, every year to every other year. So most females would skip years because there just isn't a long enough summer to build up the energy to yolk up eggs to produce them every year. But in South Carolina, females reproduced 2 times a year. They would produce 2 clutches of eggs a year. One female even produced 3 clutches, which I was blown away by. But the summer's longer, so there's lots of time to eat and accrue energy and build those reserves, right? And then they don't need to have energy reserves to get through the winter because there isn't really a winter to deal with. At least not. There's not a very long period of time where they can't eat, right? In the South. But what I thought was super fascinating when I compared the fecundity between North and South. So the females up north reproduce about every other year, but their clutch sizes are much bigger. The body size is bigger, so they have more eggs in a single clutch. In the South, this is that life history variation. The girls are smaller, so they produce fewer eggs in a clutch, but they produce multiple clutches per year. And when you take the average fecundity annually, it's the same. Just the girls up north put all their eggs in one basket every year. The girls in the South, they produce multiple baskets of fewer eggs each year. I was like, wow, that's so cool. So they achieve the same, ultimate fecundity. I was blown away by that.
That is really surprising. That's not what, given the numbers that you were telling us, I would not have expected that to be the same result. Question from the chat is just, again, what species turtle is it?
I am talking about the spotted turtle, which is a small, it's a very small black turtle with yellow polka dots all over it. It's a globally endangered species. It's endangered in Canada as well, and it's endangered in Ontario. So on all of in the United States, it has variable listing on a state-by-state basis, but it is it's a CITES listed species, meaning its trade is monitored. So illegal collection for the pet trade is one of the greatest threats to the animal. So you'll know I haven't told you really about any of the location of where I do my work, and I won't tell you because then I'd have to kill you. So I'm not going to tell you.
What is some of the complexity of working with an endangered animal in that? So like obviously you have to keep the location secret, just like if there's a new bird, right, that's endangered and you don't want to leak it because all the birders will come out and take photos and then the non-birders come out and take photos, right? But what other consideration does one have to take into account when doing this kind of work, especially with an endangered animal?
The permitting. There are layers and layers of paperwork for permits to acquire. So there's Endangered Species Act permits or Species at Risk Act, depending if you're federal or provincial. And there's different provincial agencies that require you to have a permit to handle those animals. I have to do animal care training and testing to be certified to handle those animals. So there's lots of checks and balances before I can just go out and do the work.
That's good. That's really cool to hear that there are the checks and balances in place, and also really cool that you are in fact our Jason Bourne now of the show. Again, I don't know if this has been done, but have y'all ever looked at interbreeding between the northern and the southern to see if these behaviors are innate or just a feature of detecting the environment? Because it's interesting, you were saying the southern ones, when there was that ice storm, they had behavioral differences, right, in terms of how they hid from the snow and ice. The northern ones, so it'd be really cool to know if there's anything epigenetic or, how much have they diverged, and would you call them even, at what point do you call them 2 different species, and getting into that whole hypothesis?
Well, so at one point I did have a dream, and this would have been one possible PhD idea, but was to look to see if those differences in life history were phenotypically plastic traits, or were they fixed life history traits. So I had this dream about doing like a common garden experiment or some kind of reciprocal transplant thing, but so I said earlier about for people that ask an evolutionary question, they pick a model animal to do it, like a fruit fly, because it has a 2-week generation time. Turtle generation times are like 25 to 50 years. So me and all of my students, and then their students and their students and their students could maybe address that research question. But I was trying to figure out a way if there was a laboratory version of that I could do, but I couldn't come up with a way to do it safely for the animals and that I would actually be able to get at the question. So actually at one point, I started to investigate doing it with snakes because I was going to use a captive colony of African house snakes to test these life history variation questions They have a fast life history. They hit maturity at like 1 or 2 years. They're really easy to keep in captivity, and I could manipulate the environment in the lab, right? I could do cold winters, and I could do all that. And I wrote a grant proposal to do that, but it didn't get funded.
That was a question we were going to ask, is how hard is it to get funding for these types of experiments? Because they're conservation based. And, in the US, a lot of what we hear is always slanted towards, well, how is this going to help a person? Like it's really hard to just study something because it's interesting or because you're going to learn how the world overall works, right? Without a direct connection to humans. Is that something similar that you experience in Canada, or is it a little bit more plastic in terms of what you can apply for?
So in the Canadian system, so you guys have NSF, right, to apply to for like a big research program type question. So we have NSERC, Natural Sciences and Engineering Research Council would be the sort of equivalent of your NSF. But the difference with NSERC, discovery grants, the operating grant, the point of those is to support a research program, this long-term vision with like long-term goals and short-term objectives that relate to graduate student projects. So NSF is different, right? You get a 3-year NSF. It's to do a very explicit project. It's a ton of money, but it's a very explicit. So for NSERC, they're smaller grants, but they're 5-year grants, and they're to do your program. So my research program is the ecology and conservation of freshwater turtles. So I can support that program, carving off all these little different research projects into that program and fund it with NSERC Discovery. So my research program has been consistently funded by NSERC since I started as a prof, so for 20 years. And then because the species I work on are endangered species, there are specific pots of money that are for species at risk, we call it in Ontario. So species at risk recovery and stewardship type programs through the provincial government. So I have had some success getting grants, and those are specific project focused ones for a specific endangered species. So that's how it works. So NSERC to cover the research program, and then some smaller grants, and I can leverage the NSERC funding as matching funds because these little provincial ones often require some kind of a in-kind or cash match. So and then I've also partnered with industry. So a lot of the work we also do is looking at impacts of different kinds of environmental modification on these animals. So for example, wind farms, right? Wind farms have become a really big push, a new way of green energy, better than, burning coal. And so one of the questions we've been wondering about is how do the wind turbines impact, for example, frogs? So frogs call, right? They sing to attract mates, and turbines make a lot of noise. So we wondered if the whoosh whoosh of the turbines would interfere with the frog's ability to mate, that is, call and have the females arrive. That's a tangent about frogs. But anyway, but so a lot of the time, though, my research will involve a partnership with an industry who has a specific question. So I work with one of the nuclear industries, nuclear companies in Ontario who has endangered species on their lands, and they have a mandate to respect biodiversity. So they call on me to do a research project to look at how this whichever endangered species is using the lands around their nuclear facility, and what can they do to make sure that those animals stay safe during operations. So I get grants for specific projects like that too with industry, because industry, at least here in Canada, and hopefully it continues that way, they have a mandate to respect biodiversity in much of what they do.
Then they can consult an expert such as yourself on how to actually go about that.
Well, so consult is a good word. So, there's environmental consultants, right? So industry can hire environmental consultants, and they're very expensive, and they're jacks of all trades and masters of none. Whereas they could hire a graduate student who is paid pennies, but collects incredible data focused on the research question, and the product at the end of the day is incredible, right? So this is the kind of argument I make to our industry partners. It's like, if you give me a quarter of the money you would give to that consulting company, I will get you better data, and we'll be training somebody who could then become part of your environmental team eventually, right? So that's also been a really great way to engage industry and train students in these kinds of partnerships.
That is really cool, and it really, again, clever way. Be like, look, you can either waste your money or you can actually get the good data out and have actionable items at the end of the day.
And a student gets a degree out of it.
Yeah. Everyone wins.
Right?
When you ended up finishing your PhD, how did you know, or how did you identify what you wanted your primary research question of your lab to be? And then we talked about the conservation of the turtles, but what guess what is the broad research question, and what are some of like the niche research questions you've identified over the time of running your lab that you were like really interested and passionate about? And then is there a dream something in the future that you would love to just be able to get one day, but that's kind of like that's the reach right now?
Okay. So I would say that the common thread of everything in my research program is that empirical data to inform conservation of these reptiles at risk. I would say that's the main thing. But then like within that, there's sort of the categories of that spatial ecology piece, like identifying critical habitats and seasonal activity patterns. Then there's the fundamental part about life history variation and how does that affect population dynamics. And that's a modeling heavy type of exercise to get into some Bayesian analysis with that work and look at, so for example, we have this long-term data set on snapping turtles. We have over 50 years of data. Otters came in and killed about half of the turtles during hibernation over 3 winters. So we've modeled what that did to the population. So like that, those kind of acute catastrophic events, how they impact long-lived animals like turtles. And we do a modeling heavy approach to look at those dynamics, for example. So that's the fundamental life history sort of part, even though it still informs conservation. And then the other piece, so the third piece of that is very empirical tests of recovery actions or mitigation strategies. So I talked about roads being a problem, right, for turtles, road mortality. So one of the themes of our research is to mitigate mortality of turtles and snakes on roads, you can build a fence along the side of the road that then funnels the animals to ecopassages or culverts under the road so that they can safely reconnect habitats on either side of the road, but the barrier fence keeps them off the road. So in our lab, we've tested many versions of that sort of mitigation strategy, different designs of fences, different designs of culverts, and we use game cameras and we use marker capture techniques, all kinds of different things to get at those. And we put it in a very specific kind of experimental design where we have before data and after data for the mitigations. So lots of that kind of very explicit testing of a recovery strategy. My dream thing, if I could do so, when you and I talked before, I told you about going to the Galapagos Islands. And that was I can't even put into words, bucket list. For somebody like me, that is just like the dream. So if I could go there and actually conduct a research project on those giant tortoises, that would be cool. Another one, and I got to tick off this bucket list thing too. I went to New Zealand. Have you ever heard of a tuatara?
Sounds vaguely familiar, but I can't remember what it looks like.
It looks like a lizard, but it's not. It's this very unusual, ancient reptile. And it has the weirdest life history. They're super cold tolerant. They hold, I can't remember all the they have a pineal eye. You know, if you do yoga, right? That third eye. They actually have a third eye, and it actually like has a retina in it. And their teeth are totally bizarre. They're not teeth that can come out. They're like part of their jaw. There's ancient tooth, and the males don't have penises. They actually reproduce like birds do by aligning their cloacas. Like they're just they're the last species left in this group of animals, and they're highly endangered. And there's I would love to learn more about them. I'd love to just sit with them on an island and off the coast of New Zealand and watch them and like learn about them. That would be like a total dream.
That seems like a dream experiment. Yeah, like I love the experimental designs where you can watch, and regardless of what the answer you get, it's a cool answer. And I feel like that's for all of these, honestly. Because the story that you started telling us at the beginning of our conversation, nothing was known about these turtles, right? And just through at least starting with observation and building on that, incorporating the technology, you've been able to develop this beautiful story about just what these turtles do over the course of a year, and many years of their lives where we knew nothing about this. It's really,
one of the
really
craziest, one of the craziest discoveries during my master's degree related to that was where the turtles nest. So what do you think of when you think of a turtle nest? What comes to your mind?
I always think sea turtle first. I think sand.
Exactly.
And then
okay.
I think like dirt. So like that connection for land.
Yeah. So where I did my research is in a place called Georgian Bay. It's rock outcrop, Precambrian shield, granite rock outcrop, rock barrens. The turtles nest on the rocks. So these big exposed rock outcrops that have bits of shallow pitting in them that accumulate just a little bit of soil. There's some reindeer lichen, maybe some leaf litter accumulated in there. And the turtles actually dig into that tiny little bit of dirt and put their eggs in there. And my supervisor at the time was like completely astounded. He actually wrote a little article about it in one of the newsletters saying, and Litzgus says that these turtles nest on rocks. Like, what the heck? But it was mind-boggling to me. But in hindsight, it makes a lot of sense because, again, we're talking about a northern range limit for an ectothermic animal whose summer is really short, and those eggs need a certain number of heat units to complete their embryogenesis and hatch before winter comes again, right, in the fall. So these big rock outcrops would have a lot of thermal inertia, and so the eggs, they're egg incubating hotspots, right? So the eggs would be super happy in that short window of summer that they have to meet their thermal needs so they could complete incubation. So it makes sense. But at the time, I was radio tracking. I was using those tools. I knew that this female was pregnant. So gravid is what it's called, because I could feel when you stick your fingers in the leg pockets, you can feel the outlines of the shelled eggs. So I like palpated her. I'm like, ooh, she's got shelled eggs. She's ready to go. She's got her radio transmitter. And I knew that they nested at night. I knew that from reading the literature, but I didn't know what substrate they were going to nest in. I assumed that they would nest on the edge of a wetland, like in a dirt hummock, like some dirt beside the wetland or something. So I'm radio tracking this female, and it's the middle of the night. It's dark out. I've got a headlamp on. I'm by myself, which is not a good idea. But back in the 90s, that's how we did field work. So I'm radio tracking her, and the signal is telling me she's up on the top of a rock. And I'm like, what the heck? Oh, probably a raccoon has eaten her, or somebody's, predator has dragged her up there. I'm like, oh man, this is going to suck. So I radio track, and I'm getting this signal, and the signal's kind of bouncing because now it's kind of raining a bit, and there's a bit of lightning, and I'm like, oh my God, where is this turtle? And the signal's kind of bouncing around. And I finally kind of like hung my head. I probably swore a few times. Like, I don't know where this damn turtle is. And I look down, and between my feet, there's a hummock of moss, and it goes wiggle, wiggle, wiggle. And I see the antenna of her radio transmitter sticking out from under the moss. And that's when I discovered they freaking nest on rocks. And then I'm like, okay, now I know what to look for. And so every other female after that, now I knew what to look for. They all nested on the rocks. And it's not just that species. All the other species in that area appear to nest on the rocks too. So that was like crazy.
That eureka moment too, especially after like it's raining, lightning, you're alone. The image of, oh no, a raccoon got it, just all of it coming together and then making that discovery. That must have felt really, really incredible.
It was crazy. It was like, I couldn't believe what I was seeing. Is this really happening? And interestingly, that female is the one that I told you that I brought that colleague over the rock outcrop and pointed at a turtle and said, there she is. It was her that I found nesting that night.
That's so cool. I love hearing this story, Jackie. This is it's a truly incredible system and a truly incredible body of work that you put together, and then just like all that we've learned from turtles, that feels like it would be, we feel like we should have already known this. Should have been already something that we should have known, especially if they're endangered. But it's incredible to hear the story. A quick question from the chat is, you mentioned the temperature regulation and the temperature needs that these turtles had for their survival. Is there sex determination? Do we know, is that also temperature dependent like other reptiles?
It is. So I can speak to the species I know of. So there's one species in Ontario that has genetic sex determination, and I think the other 7 are all TSD, or at least 6 of them are. And then there's different patterns. So snapping turtles so TSD is environmental sex determination, right? It's temperature dependent sex determination. And there's usually one pivotal temperature. It's usually somewhere around 28-ish. And above that, you'll get females, and below that, you'll get males. Snapping turtles actually have 2 pivotal temperatures. So you get males at the cold end and the super warm end, and females in the middle, I think. I might be wrong about that. But anyway, they have 2 pivotal temperatures, which is kind of bizarre. So they have a bit of a different version of TSD than the other species. But the spotted turtle, the one I'm talking about, does have temperature dependent sex determination.
Really cool. I wonder what the environment, like the evolutionary benefit is of having the flanking ones. Because I don't know, my brain makes more logical sense out of like, okay, this temp above this is this sex, below this is this sex. But like having it flanking and then the middle, it's like, whoa, really, really cool.
Sometimes I wonder if we've pushed the limits on all the temperatures tested in the lab, then maybe there's more subtle things happening. And some, at least a little bit more recent evidence suggests that fluctuating temperatures have a feminizing effect more so than just this cutoff of a pivotal as well, right? So there's more to the story than just the mean temperature is this, therefore the sex will be that. There's a lot more to it, I think.
Absolutely. We had a question from the chat too, is when you're testing these turtle tunnels, how does one test something like that? Is that something that you can test or you do test in the lab? lab? Is it you have to do the field testing? Is it both kind of testing? Where do you start with the materials? Where do you start with the depths? Like it's an incredible, like it makes sense to, yes, do that to preserve them, but where does one even start? Because presumably even the concrete that's poured is affecting it, and how big to make the tunnels from the chat just now. Like these fundamental questions, where do you even begin?
So sometimes the eco passages or the culverts that are used are things that are drainage culverts that are already there. Like they're engineered by the road company or whatever to be there for some kind of drainage. And then we tie in the barrier fence to funnel the animals to those. So that's sometimes what happens. There are actually companies that make eco passages specifically designed for different animals. So you can actually buy them and put them in for specific animals. Sometimes it's just, concrete box culverts that are prefabricated and they're brought to the roadside and they're put in place. Or the round, concrete culverts. Sometimes they're the HDPE, the plastic piping. So all kinds of different ones have been used and tested. And in terms of how we test whether or not they're effective, there's a few different things that we do. So we do all of our work on an active road in the field. We don't do that's not true. We've done one contrived setup for fencing. We built a bunch of different corral sort of paddock things to test fencing for snakes. But for the culverts, typically we do the test right there in situ in an active road. And we use game cameras. So we put game cameras on each end so we can monitor animals going through. The animals are marked. Sometimes there's individual differences, and so we can tell if it's an individual who entered and exited at the other end. We've also, to get at that even more closely, we've actually put antennae on each end of the culvert, and then the animals have pit tags, right? So passive integrative transponders. And that's the same, like if you have a purebred dog, your dog probably has a chip. So we put those in turtles and snakes as well. And then when the animal passes over the antenna, the information is sent to a computer so we know the animal has passed into the culvert. And then the other thing we've done is try to put that in the context of who's available in the natural environment to use the culvert. Because we want to know if that eco passage or culvert is working, some certain fraction of the population available should be actually using it to maintain gene flow and stuff, right? So sometimes we'll do a study of all the animals that live in the wetland adjacent to the road, see how many are there, and then look at what proportion of those and what complement of species available are actually using the culverts. So we put it in the context of who's available to actually be using it. And then I guess is that most, one other study we've done is we call it a willingness to utilize. Do turtles even want to go in these deep, dark tunnels? So we've set up, again, with cameras, and then we put the turtle underneath a cover, and then we hide behind a blind, like a hunting blind, and we use a pulley rope system, and we lift the cover off, and then we watch the turtle. Does it go into the eco passage or does it turn the other way? So we do a behavioral assay of how the animal interacts with the culvert as well, and try to provide motivation. So when we've done that before, we took the turtle from its home wetland on the opposite side of the road and brought it to the eco passage so that it would want to cross, like it was they know where they want to go, right? Like I said, they're tenacious. So we gave it the motivation of crossing to go back to its home wetland. And for the most part, it wasn't really a strong signal either way. What we do know is that if the fence is a great barrier, it will successfully corral the animal to that crossing structure, that eco passage, that culvert. But if there's a hole in the fence, those tenacious animals that use the same routes, they will find that hole, they will get through and get onto the unsafe side of the highway and get killed. We found that if the fencing has holes, that the mitigation fails so badly that mortality of animals actually goes up instead of down. So the mitigation made it worse because there were holes in the fence. We explicitly tested that in a very robust, comprehensive design, experimental design. That was very sad. We didn't want the animals to die, but there were holes in the fence, and we tried to get the holes fixed before we collected the data. But the agency we were working with is, nope, we're testing what's there. We're like, it's not going to work. It's going to be bad. And it was.
From the chat, what is that typical fence structure that's facilitating holes that shouldn't be there, and that would actually allow for the correct types of passages to be placed in? Or did you, I guess, find an optimal type of fencing?
Yeah, so some other people have worked on this too, not just my research group. But one of the types of barrier that we know is fantastic is a poured concrete wall. So there's a place in Florida called Paynes Prairie, which goes through this beautiful Paynes Prairie Wildlife Reserve, and animals were getting killed left, right, and center on that road. And so they did this. It's a poured concrete wall, and right into the concrete wall are the eco passages, and it goes right to the wetland. And then the road surface is on the top. and then there's a grassy edge. So it's actually a great viewing spot for wildlife. Like you can pull off on the side and then you can watch, and you're on top of this concrete barrier. That concrete barrier will stand up to everything, right? Like it's great. It's super expensive. And so a lot of, projects aren't going to want to put that kind of money forward, right? So another fencing style that we are just testing now, my graduate student actually is going to defend in 3 weeks. We just submitted her thesis a week ago. We took I was talking about those HD, I don't know if that's the right acronym, but the plastic pipes, and we cut them in half, and then we deployed them into the road so that the wetlands on this side, so it's curved, thinking that animals can't climb that. And then the road surface comes to the back of the curvature, so it's safe. I don't know. It's hard to do that without like showing you a picture. But anyway, that type of fence design seems to be half decent, but it doesn't stand up. Like we're trying to design something that stands up to snowplows and stuff too, right? And frost heave, right? Freeze thaw cycles and stuff. The fence that had the holes in it was a fence that was an afterthought. It was, what do you call that? Silt fencing, the fabric that's put in place during a construction project, for example, right? Just to keep silt from getting in wetlands and stuff. So it's a temporary style. That was used in one of our studies. It was attached to a chain link fence that was in place to keep large mammals off the road. So then they attached basically a reptile fabric fence to the bottom. But, Buddy's out there deploying the fence, smoking a cigarette, and the cigarette heater burns a hole through that fabric, and then the snakes get through, right? Like little things like that. Like it was really, we actually did a GIS analysis to show that the probability of finding dead animals close to a hole was much higher than random. Like they were getting through and then if the stitching didn't quite come to the right spot, these animals can get through anything if it's not solid, right?
And presumably it's not going to be living up to the wear and tear either, like you said, for any of the frost heaves, the snow plows, even I guess just like the daily wear and tear. I feel like
water level changes, spring melt, and then the water's over top of that fence, and it's terrible.
Yeah.
So the conclusion of that study was still good. Like we showed, don't use this fence, and that's good information, right? We very clearly showed that fence is a failure. The title of that paper is "Fence Failures Compromise Eco-Passage Effectiveness." I really like alliteration.
I mean, yeah, we do. That's why we're SciAnts streams, right? We love the alliteration. Jackie, we want to be respectful of your time. We've already gone over. We just love asking you all the questions. We were celebrating Women's History Month, highlighting incredible women in STEM. Would you be able to talk a little bit about what does it mean to be a woman in STEM and to be, an inspiration that a lot of other women can look up to in the sciences?
Oh, okay. I'm going to tell you a small story. I was at a conference a couple weeks ago, and there was a young woman professor there. I was the external examiner on her PhD defense, and she's now a Canada Research Chair, a very prestigious researcher. She stood up there and gave a talk with her 3-month-old son strapped to her, and I was almost in tears. I'm like, this is how it should be done. She's a rock star. Like, she's incredible. And there she is at the front of the room giving a talk to a room of 100 people with her baby. And I just I'm going to start crying now. I love it. That's a change, right? Like, that's certainly not how it was when I started in this gig those many years ago. I don't know what I can say. I guess I'll just go back to saying you got to run with what you love and do it.
I love it. And the image too of the mom with her kid up there on the stage is really, really powerful, and it kind of at least further highlights the hope that things are changing for the better. Jackie, thank you again so much for actually rounding out our Women's History Month on the final day with an incredible discussion. Folks in the chat are loving how much your passion was shining through and how much we've learned from you just in this hour alone. So really, really thank you for your time. Is there anything that you would like to leave us with? Any final messages? And, doors always open to have you back if ever you want to come talk with us again.
Oh, I'd love to come back and talk again. This was super fun. I really appreciate the opportunity to chat. Anytime.
Awesome. Well, thank you truly, Jackie, for your time, your insight, and your expertise in just being an incredible woman in STEM.
Thank you. All right.
Bye.
Take care. Bye.